All right, so for the project you guys are starting today on April 1st, the cell division Google Slides presentation, directions: use the following rubric to ensure that you will earn full points. You may work with one, no, it says one another typo, one other person pre-approved to complete the project, meaning some of you will not be allowed to work together just based on your previous behaviors and or experiences with that person. So make sure that you just run it by Ms. Turner, Ms. Coleman, or myself. Um, You'll be using the Google Slides and materials, 4 sets of pipe cleaners, 2 pieces of string, and 2 pieces of yarn. Now the 2 pieces of string, some of you will have ribbon because I ran out of string. However, if you're having a big issue with the ribbon, you can swap it out for some yarn. So just make sure that it's a different color yarn than what you have in your bag. So some of you will have maybe red ribbon, some of you will have gold ribbon yarn. And here's a different set because there's 40 of them all together that I made. There's 4 sets of pipe cleaners. Colors don't matter. The yarn, and then this one has string. Now, if your ribbon sucks, just come over with permission, if you have Miss Turner, and get different color yarn. Because as I was working with it, I kind of discovered it's a little bit of a pain in the butt. But if you can make it work, perfect. You're expected to use the camera on your iPad to document the different stages of the cell cycle, using only the pre-formatted Google Slides presentation shared with you in Google Classroom. So each of you will have a blank, or kind of blank, pre-formatted Google Slides presentation. As you and your partner are creating the cell cycle using your bag materials, you're going to be taking pictures of it, inserting that picture into the Google Slides, and then putting a description on it. So everyone that turns in their project will look somewhat similar. You'll have all of class today, and then it is due about 45 minutes. You'll only have 45 minutes of class time on Tuesday, April 2nd, but it will be due by the end of the day. So here is the criteria. So for full points, half points, 0 points. So overall, this project's 120 points, partially because I forgot to put metaphase in. Whoops. So I had to go back and add that. So student demonstrates a correct understanding of interface. Well, what does that mean? Student correctly depicts what interface looks like using the materials provided with the iPad camera. So obviously, no phones allowed. And student includes an accurate description of interphase. It's the same thing. I just copied and pasted it. I just changed it out. So this one is going to be for prophase, so the first stage of mitosis. Here is metaphase. Here is anaphase, starts down here, just for the formatting issues, whatever. So anaphase, telophase, and then cytokinesis. So you're demonstrating the entire cell cycle. So not just mitosis, but interphase through cytokinesis. Now for the information of where you can use to kind of help you out, in your notebook, you have this page, okay, to kind of give you an idea of what it should look like in terms of the nuclear membrane, the cell membrane, and the chromosomes. And here's also some information as well that you can use. Looking at your amoeba sisters, you should have some diagrams of what prophase, metaphase, anaphase, and telophase look like. So that's something you're going to be using as well. And then your final source of information, or I guess not final, but you can use your textbook starting on page, 392. It kind of gives you an idea. So here is interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. Now the darker circles, the kind of inner circles, are going to be your nuclear membrane. That's going to be represented by either your string, ribbon, or if need be, another piece of yarn. So in the end, you're going to have 2 brand new cells and 2 nuclear membranes and the chromosomes in there as well. The chromosomes are going to be the pipe cleaners. You don't have any spindle fibers. However, if you want to add spindle fibers, however you want to do that, you can. So you're going to have your nuclear membrane. Of course, it just went blurry. Your nuclear membrane, so you're really only going to need to use the, the string or ribbon for, let's say, interphase, prophase, and after that it kind of disappears. So you're not going to have the ribbon throughout this, but then again you'll have the ribbon back at cytokinesis, right? So that is on page 392, and there's also, oh my gosh, a little description. So this project's really, really easy if you just follow along and you stay focused. Now, when you open up your bag, depending upon what you have, this one's going to use a string because it's a little bit easier. And try to make sure that you are respectful of the materials because 2 other classes will be using them today and tomorrow, okay? So these are going to represent your chromosomes. Since they're pipe cleaners, you can bend them because eventually your chromosomes are going to be pulled apart. So you're going to have to kind of bend them to be pulling apart. Just make sure that when you're done, put them back straight. Okay. Here is this is going to be your nuclear membrane. So at the end, yes, you have 2 pieces of string. You're only going to need 1 piece at the beginning. Then your yarn should be somewhat bigger. Now, it's not a complete science. If you need to kind of make this one smaller, however, or if you need a really large, because remember at interphase, your cell is actually going to double in size. So if need be, you can make, I'll kind of pick this up a little bit. Obviously, you're not going to attach them together, but one big kind of cell. And then you have your nuclear membrane, which allows you to make it a little bit bigger. And then you're going to have your chromosomes inside. Okay. And I understand, because of size parameters, because the string or whatever, just try to make the best of it. If you need to put the 2 pieces of string or ribbon together to make your large nuclear membrane, that's perfectly fine. You guys kind of got to adapt with the materials that you have, all right, to have your chromosomes put in it like that, okay? So then you would take a picture, okay, and then you would identify. Now, when you are done, or if there's some big issue with your string or yarn, with permission, if you're in the other classroom, to come over and see me, I will have extra supplies kind of ready for you. Now, the ribbon is kind of a pain in the butt. If you have a bag with a ribbon, we ran out of string. So worst case scenario, you can always come over and get more yarn. I have more yarn than I had string. Now, these are cut kind of weird, but to the best of your ability, if it's really not working for you, if you need to have your partner take the picture, that is fine. Now, if you don't want to work with another person, because there's only 40 sets, the majority of you will have to work with partners, because let's say at most there's 28 of you or 30 of you in a class, and I only have 20 sets per class. So please don't damage them either. Please just put the supplies back. Just be responsible, guys. All right, that's it.